these target levels, are they sort of like designed and validated to sort of be kind of invariant to that? And if not, then how do you adjust for these things?
No, that's a great question. I mean, so that's where the science lies, and that's one of the things that we're trying to really do. Is, in other words, what we're trying to get at now is, that's why I wanted to show you the monkey data. So there you see the slow oscillation, and the slow oscillation corresponds to a state where the neurons are actually quite shut down. So we feel that's a very reliable marker. In other words, there's no reason to suspect that the same thing isn't happening in humans. Actually, we have human data on the same thing, actually recorded from epilepsy patients. So in that sense, the slow oscillation is a marker of a very profound state of brain shutdown. And so to answer your first question, which is yes, in other words, brain dysfunction is a real problem under anesthesia, after anesthesia. It's probably the biggest sort of clinical problem next to like nausea and vomiting that patients have. And it's highly prevalent in older patients. And one of the reasons is that, you know, most anesthesiologists don't use, I mean, think about this. We're obliged to monitor everything: heart rate, blood pressure, body temperature, oxygenation, CO2. We're not obliged to measure your brain, but the drugs are acting in the brain. And so, and then one final point about this: of all the things that the EEG is used for, whether it's to track sleep, patients in coma, do cognitive studies, the signal under anesthesia is the most highly structured and has the highest signal to noise ratio. And you saw it in the video. You saw when the drug took effect, you saw how the artifacts went away. So if we can't use EEG in anesthesia, then we can't use it anywhere, because we've got the cleanest signal. So, and there's work to be done on saying, what does that signal on the top of the head mean in terms of how the circuits are turning off or changing dynamically in the brain? But I think that's a very, very tractable research question.
So a related question, I guess, would you sort of like build in an additional constraint or a regularizer or something in your optimization problem that basically says, let's really go for sort of like the least dose necessary to sort of like hit your targets?
Yeah, I mean, I think that's totally feasible. And so we're, you know, we're at different stages with this. So what I showed you what we've done with the rats. Now we've got something going on with the non-human primates. And the real test in the non-human primates is going to be with EEG. Like what I was showing you there was with local field potential. So those are electrodes actually inside the brain. Mm-hmm. So now what we need to do is do the same thing with surface EEG, because that's what's going to be translate to humans, if you see what I'm saying. But yes, I mean, you would want to be able to use as little as possible to achieve the states. And I can tell you, when I use these sort of physiologic principles to actually dose my anesthetic, I'm guiding manually, I give much less anesthesia to patients, and I can have them wake up with clear heads.
Yeah.
So if I can train the computer to do this, the computer can watch the patient better than I can. If it's changing the dose every 20 seconds and I'm changing it every 20 or 30 minutes, the gains are enormous.
I really like that story. It just goes all the way from fundamental neuroscience all the way to something with profound clinical implications.
Yeah, it is. Yeah.
Karsten, so a lot of your work really, I think, I'm guessing that it's sort of like the work that you presented partly grew out of like starting this like sort of big multi-center site, where one of the first steps you really need to take is to just really take a really gimlet-eyed view of the literature and figure out, okay, what's going on here and how do we sort of like avoid some of the pitfalls. And by the way, I think like Karsten's talk is a great one for authors, readers, and reviewers, because it, you know, one of the key things is that how exactly you formulate your learning problem and your evaluations matter a lot. So one of the listeners had, one of our conference members had a pretty interesting question, Karsten. Like, how sensitive are like some of your final conclusions to the way that you harmonized the data sets? And this sort of like goes toward like sort of like the validity of like the formulation of the technical problem that you're really solving. So like another way you could ask the question is like, well, you harmonize the data, and I assume that means that like you picked a particular way of operationalizing a definition of sepsis and you standardized on that. And that's great. But presumably, these 22 papers that you surveyed, they also had some, justification for operationalizing or picking the sepsis definition that they did and operationalizing it the way that they did. And like, are your conclusions sensitive to those choices, or do you feel that like, actually this is the best way to formulate the problem, and I'm going to, just back it up with this data?
Yeah, thank you very much for these comments and this question. So certainly all these results are always sensitive to particular choices you have made. But I think the key challenge is to make the same choices across different data sets. I mean, the data sets are, by the way they were collected, by the different standards in these different countries and different ICUs, they are inherently different. So the very least we can do as machine learners is to try to define the label, the endpoint, in a systematic, uniform fashion across these different data sets. And that's what we tried in this study. There were huge obstacles in that, no doubt. That's actually, in my opinion, the reason why this hasn't been done at large scale so far. So sepsis 3 includes this SOFA score increase, which includes a lot of variables that have to be measured for each patient. And there's also the suspicion of infection that is part of the sepsis 3 definition, for which not all aspects and inherent data are available in all of these ICU data sets. So we tried to use the same definition across the different data sets, and where the data was not available, we tried to find a very good, very close proxy and to show statistically that this is a very good proxy. So in short, yes, this is a major challenge to make the results valid and comparable, but that's exactly the challenge we try to tackle and to address in our work here. We did our best to circumvent this problem. Yeah.
So Karsten, one more sort of like neuro technical question. Actually, this is more sort of a speculative question. So Jim Fackler, who's an intensivist, a pediatric intensivist at Johns Hopkins, and one of our founders of this conference, he asked a question about, so there's actually at least one commercial system that sort of like aims to do sepsis prediction in the ICU, and it wasn't very good at it, or at least it didn't seem to lead to improved outcomes. Do you have any speculation about what went wrong there?
I don't have speculation. This is the Wong et al. Paper that I mentioned during my talk. Yeah, so I don't want to speculate now on the reasons of failure. What I think is that such systems can now, after our study, be much easier validated across a number of different external data sets. So what I know is for that study is that it was first successfully validated in one state in the US, or one university in the US, and then it wasn't very well validated in another state, and only like around 63, I think, percent area under the ROC curve, and actually before that it was already commercially deployed. So now with our harmonized ICU data set, you could do at least a statistical validation in 3 different countries in 4 different data sets before you commercially deploy the system. And I think that's a significant step for the field to be able to do that, and to do it rather easy.
Yeah, that's great to hear. So this is going to be a little bit of a shift away from sort of like narrowly technical questions, and I think like something that struck me about both of these lines of work is that they are, in your case, Karsten, it's a little bit like sort of at the early stages, but it's a huge effort, like involving, like it's a big multi-site effort. And it's going to extend far beyond potentially the sort of like developing and validating, methods and models, et cetera, to potentially like, trying to figure out, okay, what does it take to operationalize and actually achieve some benefit from using these in ICUs? So if you look at like systems that have actually been demonstrated to provide some clinical benefit, for instance, like Kaiser has like a an early warning, inpatient early warning system. But most of the work wasn't really centered on the, like, at least as measured by publications, a lot of the work wasn't really about like a marginally fancier model or something. It was really about how you operationalize things and adjust workflows, managing change so that people, you know, in the clinic who are already very busy don't revolt. And for you, Emery, like you started like, you know, this line of work that eventually ended up resulting in a closed loop system for administering or controlling administration of anesthetic. That spanned at least a decade. And a couple of things that I'm kind of hoping you could do is give, especially some grad students, a little bit of perspective on how long and how to stay motivated over a really long arc of work, because these are both sort of like pretty, at least they're ending up in very translational work. And I think a lot of the people who come to a conference like this, they're on some level very motivated by the idea that like eventually something that they're going to do is actually going to provide some clinical benefit. But the road to that is extremely long sometimes. And it usually involves all sorts of collaborations. So Karsten, you're going to have to, like, you're forging a huge collaboration across, you know, like, as you said, every research hospitals, ICU in Switzerland and beyond. Emery, you mentioned you have a long-standing collaboration with a mathematician, Nancy Capel. And I'm wondering if you could comment again about, like, well, how do you forge and maintain those relationships? Because that's sort of one of the things that this conference has always sort of like strived to do. It's to bring in or bring together people with very different sort of like domains of expertise. But it's pretty difficult, and it's really hard to measure how successful you've been with that, especially after you sort of like go beyond like sort of like easy to measure things like co-authorship and stuff like that, and really start to get into like, you know, the sort of like the nitty gritty of like actually bringing things to clinic. And by the way, I'm kind of hoping that this is more of a dialogue between the two of you, because
Emery, do you want to go first, please?
Yeah, I'll start off. And I think the most important thing, Ken, is that the things we're doing, they're fun. They really are fun. I mean, you go into the operating room, you see these signals, and you go, wow, I mean, this is like amazing. And then you start looking at the properties of them. You can see the spectral properties very clearly. And then, like you said, we talk to somebody like Nancy, and we just ask the question, if this is the anatomy, and these are the signals we're seeing, how can the brain produce these signals? How might they come about? And then you can say, all right, now that you see, I have some idea how they might come about, can you do experiments to validate them? Can you put electrodes in different places and show that they're there, right? And I think that, so those are, I think those are, at least to me anyway, I find them very, very fascinating questions. I mean, it helps to give us some insight into the way the brain is working, as I was suggesting in my last comment, insight into the way the brain is working from a perspective that we don't typically think about. We don't think about using anesthesia to study how the brain might be functioning, how the various parts function. But in order to really understand what anesthesia is doing, that's exactly what you have to do. You have to really start to understand how the various parts work. And in terms of the length of time, I think that even though it's taken a while, we found out interesting things along the way. I mean, I've given you like a quick synopsis of it, but you know, there are large chunks there that, you know, that have come to fruition, and we've come to understand, you know, I think we've come to understand very, very clearly, and they've laid the foundation for even more clear thinking. And in particular, like with the various brain states and what they mean, you know, I think now we're getting into understanding exactly how the circuits are altered to generate, you know, dynamics like this. And so this then has implications not only for things like anesthesia, but also for coma, for understanding sleep better, for understanding brain states like depression. So what you start to realize is you have insight not only into anesthesia, but you have insight into the neuroscience of the brain from a unique perspective.
Yeah, I find this highly fascinating, Emery. It's a very exciting approach to study the brain. I was wondering, when listening to your talk, you showed these different examples of persons of different age and how their oscillation patterns differed. Have you tried to turn it upside down and to take the patterns and predict the age of a person from that?
Well, it turns out you probably can. I mean, because what I was showing you with them slowing down, I mean, there's going to be some between person variation. Remember that 56-year-old guy who looked like the 81-year-old lady?
Yeah.
There's also a 57-year-old guy who looks like a 30-year-old. So there is between person, there is between person variation. But the thing is, if you know the drug, right? So I could trick you by saying, okay, I show you a pattern that looks like, a nice slow oscillation from an old person, but it's not propofol, it's a different drug.
Yep.
So what you have to do is, if you know the drug, if you know the, you know roughly the dosing range, you can make fairly clean inferences. You can make fairly clean inferences about, the brain state of the patient. And that's what's underexploited to this point. In fact, the automatic algorithms that are working in operating rooms today don't take into account the drug you're being given. They don't take into account the patient's age. They don't take into account the dose. In other words, if they see an oscillatory pattern, no matter where it comes from, they make an inference about the level of unconsciousness, which isn't correct. Because if you take a drug like dexmedetomidine and you see the slow oscillations, I could go to that patient and wake them up. If you see the same slow oscillation with propofol, there's no way in the world you're going to wake them up.
I see. And do you think you can make predictions about brain health or even future brain health based on these patterns, like neurodegenerative diseases or similar tasks?
So this is something which I think is very important to point out, because remember, before there was prediction, there was just description, right? And so for the moment, let's not miss the opportunity to just describe what's going on.
Yep.
In other words, I think that, just getting characterized how the oscillations change as a function of age, and then going back and doing some neurophys to understand why that's the case, that's going to be like extremely useful. And, could we, eventually do some predictions? Yeah, I think you could create a brain stress test. So for example, I could give you a little bit of propofol and then see where your oscillations are. Because I've seen people, like I had a 33-year-old gentleman who I was talking to him, I was taking his history. He's just going to have a fairly small procedure. And I couldn't put my finger on it, but something wasn't right. There was something in the way his affect was just seeming a little off. And so I always ask my patients about their physical activity. I want to know how healthy they are. And I said, do you play sports? He goes, oh, yeah. He says, I do exercise. He said, yeah, I do. Like right now, I just run. He says, but I used to play rugby. And then when I looked at his EEG under anesthesia, he looked more like that 56-year-old, and he was only 33. So there's something there. It has to be studied in detail. But I think there's something there. But I think the high payoff question right now is just using these patterns to infer anesthetic state.
Yep.
And I think if we can refine them enough to do that, that's going to be beneficial to a lot of people in a lot of places.
And then the prediction is downstream. It's often basic research, I guess. Downstream basic research that could follow up on.
Yeah, I mean, so but remember, for the closed loop control, you have to do very accurate prediction.
Yeah.
I mean, that's like real time. So it's not like, let me see what's going to happen 3 days from now. It's like what's going to happen 3 seconds from now.
Yep. Yeah, very impressive.
Oh, thank you. So I'm curious, like with the algorithms for the prediction of sepsis, how would you see a clinician? I'm an ICU physician, and I've got this algorithm. How do I use it? In other words, it's a cool result, but then when I'm taking care of patients, how should I plug your algorithm into my sort of day-to-day calculus in taking care of patients?
So thank you for this question. That's a very relevant question. So we usually model, and we and actually most of the field, if not all of the field, models this as a binary classification task to mimic an alarm system, that an alarm goes off. So if the prediction goes to 1, then an alarm at the bedside in the ICU goes off. So it's not a time to event prediction or other statistical techniques that you could also use. We really think of this as an alarm that's going off. And of course, like the big problem in practice is alarm fatigue. If that alarm goes off all the time or goes off 100 times, then next to one patient's bed, then no one will pay attention to this anymore. So we need to have a high precision in these alarms as well. And actually, that's one thing we also try to do very systematically in this paper on the harmonized ICU data set. So we only evaluate the accuracy of the first alarm that's being raised. Unlike other studies, so we don't pool over hundreds of predictions and then say were like 50 of them correct and 50 not. So we don't do it like this, which would cause a huge alarm fatigue. In practice, we go for the first alarm. We focus on the first alarm. Is the first alarm correct? And how early does it happen relative to this onset of sepsis? And then, of course, this is then the open question: what would the physician really do in that moment? What would the physician really do if there is an alarm saying in 4 hours from now this patient will develop sepsis? I think this we just have to put into the clinic to see how helpful this really is. Then in practice, that's what we are planning to do. So we are working with the different university hospitals in Switzerland. We know their data warehouses now from collecting all of these electronic health records, and well, one of the next steps is to bring it back to 2 of these ICUs we are planning at the moment, and to see whether having such an alarm next to the patient improves the outcomes of the patient, like reduces mortality, reduces the length of stay, for example. So that's how we want to bring this innovation back to the bedside, back to the patient.
Oh, that's cool. And then just one sort of follow-on question: what about the physiologic insights? So you have this model that predicts sepsis, and let's say it works very accurately. You have very respectable areas under the operating on the ROC curve. Do you learn anything about the physiology of the sepsis that you're seeing, or the epidemiology of it, that can again be another way that this could help practitioners sort of take care of patients better?
Yeah, so we can do a feature analysis, which features contribute most to our model. So we did a Shapley analysis in our paper. It's, if you go to if the interested audience goes to this paper, it's figure 5. We do a Shapley analysis and say which variables contribute most to an accurate prediction. So this is not at a level yet that we could like make a mechanistic model that the interplay of these 5 variables here is related to the development of sepsis, but we can tell which variables are more or less important for correctly predicting that sepsis will occur soon. So at this level, we can do it. We can say this is the ranking of variables that contributes most to these predictions. But
and what would you say they are at the moment?
Yeah. Excuse me?
No, what would you say they are at the moment? Do you have a sense of what they may be?
So I can give you the highest ranking ones. So the mean arterial pressure, the heart rate, diastolic blood pressure, urine output, oxygen saturation. These are the top 5 variables at the moment. Yeah, I think that heart rate is involved in sepsis, or the changes in heart rate are indicative of sepsis is actually known from the literature. So it's not totally surprising what we see here at the top of the feature list. But investigating this further, more systematically, coming up with ultimately mechanistic models, that's certainly on the future work list here in this field.
Karsten, so like there was a question that I'm going to try to fold it into this conversation. So I think you were mostly showing area under an ROC curve, but it does seem like, well, maybe you should be using something more like a precision recall or a mean precision measure there.
Yeah.
But related, and maybe the bigger question is sort of centered on like the and this is all about like evaluation. part of the reason people are interested in like, well, how much like lead time do you get is that like I've occasionally heard intensivists say, look, these models aren't telling me something that like an experienced, intensivist or, ICU nurse can kind of like look at the patient, because the ICU is a very, high, there's a lot of interactions with patients, and often the staff actually has a pretty good sense. And if you're not predicting things which are in some sense like a little bit surprising well ahead of like when it's kind of suspected or obvious to the clinicians themselves, it's, sometimes the questions are like, well, what is this adding that I don't already know? And what's your response to that sort of question?
Okay, so the two questions. First, the ROC values. So I did not only present ROC values, I also had this precision recall plot versus earliness. So we also look at precision and recall. I think for comparing the different internal and external validation performances, our ROC is fine because it gives us a way to compare across these different data sets, and it should be robust to the prevalence that may also vary between the different data sets. Now we also look at precision and recall. We fixed recall at 80%, like correctly discovering 4 out of 5 sepsis cases, and then see how the precision is, and that goes up to 50% in the internal validation, and on the Amsterdam dataset, that's our prime example, and to around 35% in the external validation. So we have also rather high levels of precision and recall here in this system, and we have earliness with respect to the sepsis onset. So I think it's not only limited to our ROC here, and we are not hiding anything in our ROC, it's also in the other metrics. The predictions are doing really well. Now the second question, the comparison to a nurse, like a practitioner, is an experienced doctor or experienced nurse maybe as good at detecting sepsis as we are? I can't give you a definite answer to that yet. I think that this is something we have to find out in the clinical validation, and that's like really far up on my to-do list to do this with intensive care units in Switzerland, like to see whether the algorithm can really help them and do something they wouldn't have done without the system. Yeah.
Cool, thank you very much. Professor Brown, a quick question from you. Oh, this is from one of our organizers. So do you think that like variable response to anesthetic could sort of like help identify risk of Alzheimer's disease or other dementias?
I honestly don't know. I wouldn't. I think what it can tell us about is something about aging, and that I feel pretty comfortable about, because we've taken time to sort of map out why the patterns look the way they do as a function of age, and it has to do with like which parts of the circuits have either developed or which parts of the circuits have degenerated. But you can certainly degenerate with age, and whether there's a specific signature with something like Alzheimer's, I would doubt that. I think that would be like a long run for a short slide. But something that could tell you something about whether or not the brain was frail or the brain was aging faster than it might. Like, for example, if you have autistic children, the brains of the EEG under anesthesia of autistic children look about between 15 to 20 years older than their actual age. And so something about aging, I think, yes, because that has to do with how the circuits are hooked up and how well they're working. But for Alzheimer's, I wouldn't rush off and do that first thing.
Thank you. I think we are at the end of the QA session. Did you guys have any sort of like parting remarks that you wanted to make, or questions that you want to ask yourselves?
No, mostly just thank you for this opportunity. I mean, it's a phenomenal experience for me. I mean, I really appreciate it. So thank you so much.
I fully agree. I thank you. It was a great opportunity here to watch Emery's talk. Very impressive work, and
likewise.
Discussion session. Thanks for the opportunity to present my work also.
Thank you.
Thank you.
Okay, well, it was certainly a pleasure and a privilege to have both of you speak.